Okay, so we'll be talking about what is the definition of a compound. Okay, that will basically be two different elements or more that will be connected by one of two bonds, either a covalent bond or an ionic bond. Do you know the difference between the two types of bonds? Okay. So the case of covalent bond would be the one that's essentially between two non-metal elements, while the ionic bond will be between a metal and a non-metal. Okay. A good example of a covalent bond would be basically H2. Okay, H2O, N2, CO2, O2. And a good example of the ionic bond is going to be NaCl or CaCl2. Now, when you look at the covalent bond, you essentially can classify it into two different types. Either a polar covalent bond or a nonpolar covalent bond. Okay? Okay, so in the case of the polar covalent bond, we're essentially discussing that there's going to be unequal sharing of electrons between the two atoms, where one of them is going to have a bigger hold on the electrons and is going to be given the symbol this, and the other one is going to have lesser hold on the electrons with this symbol. So the one with the negative symbol is going to be more electronegative, meaning that it's going to have more hold on the electrons, but the positively charged one, it's going to be less electronegative, so it's going to have less hold on the electrons. And a very good example here is going to be water. So water, or H2O, okay, it's written like this, and the O is famous for being highly electronegative. Therefore, the electrons want to aggregate more on the oxygen, given it this sign. But the hydrogens are not so acquainted with the electrons. They don't like the electrons that much. And they're going to be willing to give them away to the oxygen. Now, of course, there is no loss or gain of any electrons. There is only sharing of electrons. Okay, but this is going to be sharing of an unequal nature. So we're going to say unequal sharing. They're still sharing, but simply they're unequally sharing. Okay. Now, let's talk about nonpolar covalent bond. So that would be the case of bond. That would be the case of two atoms of the same exact element, like the N2 or the H2 or the O2. Okay. Sometimes you would have a polar covalent bond, but the molecule would be nonpolar. The reason for this is beyond the course. It's more for chemistry. But for bio, just know the example. We have one example, which is CO2. Okay? So CO2 has a polar covalent bond, yet the molecule is going to be nonpolar. And finally, we must list for you what's going to be polar molecule versus nonpolar molecule. So a polar molecule that would often come on the AP is going to be water, is going to be glucose. We have certain molecules called amino acids that come in two types. They can be polar or nonpolar, polar amino acids. These are the most common ones, okay? We also have peptide hormones, which also tend to be polar.
Do we need to memorize these for the AP, like to list them down?
We do have to list them down, yes. Okay. We also have some hormones that you don't have to know the names of. The exam will tell you their names. That will also be polar, okay? How about nonpolar molecules? Things like CO2, nonpolar amino acids, and certain hormones called steroid hormones. Okay. So now there is something else that you guys have to know, which is called hydrophilic versus hydrophobic. If it's hydrophilic, it's one of two things, either polar or ions. But if it's hydrophobic, it can just be one thing, which is nonpolar. And of course, the chemistry of these is going to be super important because ultimately we have to talk about the cell membrane. You see, every cell is going to be surrounded by a membrane. Now, this membrane is made of two layers. One layer looks like this, while the other layer looks like this. And imagine now hundreds or thousands of these layered up and protecting the cell from the inside, from the outside, and also regulating what's able to go in and also what's able to go out. So what's the chemistry behind the cell membrane? The interior of the cell membrane, we call this part the interior of the cell membrane, is made of those tails that you guys see. And those tails are going to be nonpolar or hydrophobic. So we're going to call these tails hydrophobic tails. Okay. So this interior will determine what's able to go in and go out with ease or without ease. So because here we have the interior is made of hydrophobic tails, they will allow hydrophobic substances to move with ease, and they will give a hard time to hydrophilic substances. So hydrophilic substances, which are either polar or ionic, can move, but they will need some help that we'll discuss in a different chapter.
What are these, the heads of the tail? Are they also hydrophobic?
They're hydrophilic.
Philic?
Okay. Yep. So this is the phospholipid, right? That comes in two layers. This head part is the hydrophilic part.
This is the one that lets hydrophilic substances in?
No, that's a different thing. It's called a channel. Yeah. And these tails are going to be hydrophobic. All right. Okay, now it's time to talk about something called weak chemical interactions, okay, which are not covalent bonds nor ionic bonds. They're going to be something else, okay? We have the most famous of these is going to be hydrogen bonding. Followed by van der Waals forces. Each one is famous for a certain type. So if we're discussing water, that's going to be hydrogen bonding. Okay. If we're discussing van der Waals forces, that's going to be for something that is going to be nonpolar. Both of which are going to be super important, with hydrogen bonding being the stronger ones in the mix.
Okay? Hydrogen bond is polar?
Hydrogen bonding is for polar substances. Not any polar though. They have to have certain elements, more for chemistry, but water is very famous for hydrogen bonding. Ammonia is also famous for hydrogen bonding. Okay. Here we have a hydrogen bonding forming between the hydrogen of one atom and the nitrogen of a different atom. Because here's how it works. So here is water, and here's ammonia. Ammonia is going to be NH3. Now, in water, the oxygen is going to be partially negative, but the hydrogen is partially positive. While in ammonia, the nitrogen is partially negative, and the hydrogen is partially positive. And a hydrogen bond can form between a partial negative of one molecule and a partial positive of the other molecule. And you must also always represent it in the form of dotted lines, never a straight line. So a hydrogen bond can now form between this H and this N, for example. Or it can form between this O and this H.
So between it always has to be positive and negative?
Exactly.
Never positive to positive?
Never. That's not going to happen. Okay?
Opposite.
It has to be opposite charges, yes. Okay? Now moving on to van der Waals interactions. Like I told you earlier, they're mainly reserved for non-polar substances. They're going to be weaker than hydrogen bond, but they still matter. For example, this animal here, this gecko is able to stick on the wall by relying on these van der Waals interactions. Okay. So the polarity of the cell membrane is going to be very important. Here we have a cell membrane with a bilayer essentially, and this yellow part is going to be the hydrophobic part.
It's the tails, right?
The tails, exactly. And so now, if a hormone like endorphin wants to enter the cell, we have to ask about its chemistry. Is it hydrophilic or is it hydrophobic? As long as you see the hormone only being able to bind to a receptor and not being able to enter the cell, this tells you that your hormone is going to be the opposite chemistry.
Hydrophilic?
Hydrophilic. So in that case, it's not allowed to go inside. Now, some hydrophilic things will go inside, but some can never. They're simply too large, like this hormone. So instead, it will simply bind to a receptor that's going to be on the cell membrane. Okay?
What are receptors?
They're things that are, look, first of all, they're made of proteins. And because this hormone can never enter the cell, it will simply bind to the receptor, and this receptor will then communicate the message to the inside of the cell.
Okay,
so the hormone can stay outside.
Okay.
The hormone will stay outside. It will never enter, but its message will enter through the receptor. Okay, let's talk about photosynthesis for a bit. But of course, there's going to be a different chapter reserved for photosynthesis. It's about basically how the plant or the algae, they make food for themselves. You have to have two reactants, which is the carbon dioxide and the water. And of course, you must have an input of sunlight, and your products will be glucose and oxygen gas. Okay. Now,
we're supposed to know that reaction, right? If they ask for how is photosynthesis, we need to know the reaction.
that is if you're lucky, but basically you have to know it in context. So knowledge-wise, memorization-wise, little, but concept-wise important. Yes, of course you have to know it's water and CO2, but the exact numbers, no.
Okay, so we don't need to know it's 6 CO2.
No, they'll never ask you that.
We just need to know it's carbon dioxide and water,
Exactly.
sunlight, and then as a product you get oxygen and glucose.
Exactly correct. Yep. Now, a reaction rate can be affected by multiple things. One of those things is going to be the concentration of a reactant. If you plan on making the rate of the reaction higher, then I recommend that you increase the concentration of the reactants, because by doing so, they will then collide with each other more, and that will give you more products. Okay? When they collide with each other more chance of reacting, and therefore more products will form. Okay? Now, speaking of water, so whenever you see dots forming between two atoms of two molecules, those dots will represent what?
Bond?
The hydrogen bond. As long as there's hydrogen involved with some other element, that could be either oxygen or fluorine or nitrogen, it's qualified as a hydrogen bond. Here we have an oxygen that's partially negative interacting with a hydrogen that's partially positive. And that's going to be a hydrogen bond. This will keep the water molecules close to each other. Now look at these two pictures. Here we have liquid water, and here we have an ice. What's happening? In the case of liquid water, you see that the molecules are closer to each other. But in the case of ice, they're what? They have spread out. They're farther from each other. That's actually a fact. So in the case of ice, the water molecules are going to be farther from each other, which will then make them occupy more volume, which will then reduce their density, which is obvious in real life. If right now you put a bottle of water in the fridge, or let's say the freezer, maybe for 2 hours, and then come back, it's going to be, the water will take up more space, right? Possibly popping the cap. So in the case of the ice, the volume occupied by the frozen water molecules is going to be more, bringing that density down, which is why ice will be able to float on the surface of the water.
But isn't ice partially solid?
It's actually all solid.
Yeah, so how is the particles further from each other?
They're further because they occupy more space. And let me tell you exactly why. So in the case of a water molecule that's in liquid form, the angle between the water molecules is going to be 104.5 degrees. But if we're discussing ice, which means a solid water, the angle is going to jump to 109.5 degrees. This increase per molecule will result in overall a larger volume. So right now, this water in a bottle will simply expand more if it's ice form. It's going to occupy more space. It can even break glass bottles. And density is equal to mass over the volume. So when the volume goes up, the density will then decrease, which makes the ice float on the water. But the ice floating is not just a nice thing to look at. It's also important for nature, because a floating ice is able to then insulate the water below. Because look, if we have a river, right? Let's say you have a frozen river. Not the entire river is frozen, just the top surface of it. That means that any animals that are living on the inside, below the ice, are able to live, because the ice is not going to sink and kill them, number one. Number two, If it's outside the ice, let's say negative 10 Celsius, below the ice could be more than 0 Celsius, maybe a 4 Celsius. The animals will not freeze to death. So the ice will insulate them and also allow them to live when they're below it. This is all thanks to hydrogen bonding, by the way. So what are some other properties of having a hydrogen bond in a water? Now, because of hydrogen bond, water molecules are able to stick to each other. We call this cohesion. That means that they will stay close to each other. Okay? Now, why is that important? For two things. When they stay close to each other, this will help form a layer on top of the water. So some insects will then use that layer to walk on the water, okay, which by the way is called surface tension. So right now, if you take an insect that can walk on water, take the same insect on oil, it's gonna sink because oil doesn't have any surface tension. How about the other benefit? Right now in your house, if you want to take water all the way to the second floor, you have to have that blue thing in the outside called a motor, right? Or a motor. But a plant doesn't have to have that. It can be 10 meters in height and has the water uploaded to the top leaves. Partially thanks to cohesion. Because the water molecules will stick to each other. They're going to be moving as units with each other. Up against gravity. Okay? Which will then help the water go all the way from the root to the top leaves.
So because of cohesion, the water travels
multiple things. One of them is cohesion, yes. Okay? And all of these are caused by hydrogen bonding. And here's an example of the application of surface tension. This spider is then able to walk on the water. Surface tension is the result of cohesion, and cohesion is the result of hydrogen bonding. Okay. Yes, cohesion is going to contribute in the transportation of water against gravity. Okay, against gravity. But cohesion alone is not the answer. You must also work with two other things. One of them is going to be adhesion, which is the ability of the water to cling to the surface. To the surface of who? The surface of the stems from the inside. So cohesion plus adhesion work together to move the water against gravity. Cohesion is the sticking of the water molecules to each other. Adhesion is the sticking of water molecules to the walls of the stem from the inside. Both caused by hydrogen bonding. And of course, there is one more thing that's going to help them, which is called transpiration, which is the evaporation of the water from the leaves. By doing so, water is going to be pulling the water that's below it upward. So as the water molecule from the leaves go away by evaporation, it will then cause the other water molecules to get pulled toward it. Okay?
What does that mean?
That means that if we have, it's like having a queue, right? Here's the leaf. This water molecule here evaporates. Now it's going to leave an empty spot. So now it's going to be dragging the waiting water molecule. It evaporates, it will drag the other water molecules. So they keep dragging each other upward thanks to transpiration. So having sunlight is important, not because of only food making, but also because transpiration does help in pulling the water upward from the roots.
So we could just write that as like a definition.
They will never ask you to define it, but yeah, for context-wise, transpiration means the evaporation of the water from the leaves, which will help in the upward movement of this water. Mm-hmm. Adhesion will be also helpful in resisting the pull of the gravity by these water molecules sticking to the walls of the container. And of course, cohesion is when they will bond to each other by hydrogen bonding and also move against gravity. Okay, water is also going to be helpful in the moderation of temperature. That basically means what? Water can act as a heat sink. So the heat of the weather can sink into the water. That's why people who live, next to the beach are going to have a better weather than those who live in the dry land. Okay, it's going to be effective as a heat bank, basically. Okay. Water is going to have a high specific heat capacity thanks to hydrogen bonding, which basically means what? It's going to be hard to change the temperature of water. It's going to be hard to make it higher. It's going to be hard to make it lower. Okay, which can be explained if you want to heat up some soup in the microwave. You take the bowl of the soup in the microwave, and let's say after 2 minutes the bowl becomes super hot to touch, but the soup is still not that warm. That's because of water's high specific heat capacity. Okay, which by the way is a great thing, because right now if it's outside, let's say it's 42 Celsius, that's going to be very hot outside, but your body is still at 37 Celsius thanks to the water doing most of the work. Or let's say it's very cold, your body is still 37 thanks to water having a high specific heat capacity. Okay, so water is going to be very hard to change its temperature compared to other liquids, basically. Thanks to what? Thanks to hydrogen bonding, because hydrogen bonds are going to be harder to break. They require a lot of energy to break them.
That's why in summer the ocean doesn't get that hot.
Yes. That's because water just refuses to change. Yes.
Even in direct sunlight.
Yeah, even. Like right now, if you go to a pool's gonna feel cold when the weather is like hot outside, right? Okay. There is something called evaporative cooling, which basically means that your body will cool down upon sweating. Sweating is actually very important because if you don't do it, you're gonna overheat. You might have a heat shock or a heat stroke. So sweating is going to lower your body temperature down. And that's obviously a good thing, right? So sweating, how does it work? Your body's heat is going to be used to evaporate the sweat. When that happens, that's going to bring your body heat down. And of course, water, thanks to hydrogen bonding, has a high heat of vaporization. That means it's going to be taking lots of energy to actually vaporize and leave your body. And the energy comes from where? From your body. So your body cools down upon sweating, upon evaporation of the sweat. Okay? And that's called evaporative cooling. And of course, if you go to a humid area, it's going to be hard for the water to actually evaporate. Humidity fights evaporation. So if it's a humid area, you're not going to be feeling very comfortable because when you sweat, the sweat stays on your body, does not evaporate at all. So you're going to stay feeling, warm. It's not going to feel too good. Okay, you're going to feel discomfort basically.
But isn't evaporation feel like... Wait.
Evaporation can only happen if the air is dry, not humid. So if you go to a humid area, you will sweat, but you will always feel wet.
Yeah.
There is no evaporation. Okay, we have an acidic environment, we also have a basic environment. If it's acidic, that basically means that we have lots of H plus around, which also means that the pH has to be less than 7. If it's basic, that means we have lots of OH minus around, which makes the pH more than 7. Okay? Okay, now, having CO2 in the air is not a good thing for the oceans, because having CO2 in the air, which is the result of the burning of fossil fuels, which are found in cars and planes and other things, this CO2 will end up in the water actually. So CO2 will react with the water of the ocean, forming carbonic acid. But carbonic acid will now dissociate into two things, into hydrogen ion and also into bicarbonate. This hydrogen ion will then react with the negatively charged carbonate. So this guy here is called a carbonate ion. So the H plus will then interact with the carbonate ion. What's the source of this carbonate ion? The source of it is the layer of this ecosystem under the water that's called a coral reef. So this is like a forest, but simply under the water, and the walls of it are made of carbonate. But this carbonate, when it sees the hydrogen ion, it will go crazy and it's going to leave this shell and it's going to react with it instead. So essentially, having CO2 dumped into the ocean will then make this coral reef lose its shell slowly, and it's going to lose mass over time, and this ecosystem will die over time.
So what is H2CO3?
It's the acid that forms in the water because of the CO2 in the air reacting with the water of the ocean. And this acid, H2CO3, will then break down into two things, into a hydrogen ion and also into a bicarbonate. This hydrogen ion, which is positively charged, will then go and react with the carbonate, whose source is the decaying coral reef.
So this is bad for the ocean.
It is very bad. This phenomenon is called ocean acidification. It's a bad thing. It's not a good thing. Okay? So CO2 is able to cause ocean acidification. That means the oceans become acidic, which is not a good thing for the health of the ecosystem of the coral reef. So, how did Earth form? The prevalent theory in science, was formulated by a scientist whose name was Stanley Miller. This scientist back in 1953 concluded that early Earth, so before humans were born or any animal was born, formed because of early Earth only. That basically means that there were special conditions on early Earth that we don't have today, and those conditions were responsible for the formation of life on early Earth back then. So what are those conditions that do not exist today? Conditions like having lots of ammonia in the air, which by the way is very toxic, having lots of H2 in the air, lots of CH4, lots of water vapor, having lots of lightning.
What is CH4?
It's called a methane gas, which is also poisonous, but again, back then there was no life yet. So these poisonous things and these abundant things combined in a way that was able to somehow form a basic amino acid. But again, those conditions only existed then. We don't have those conditions today. And those conditions together were able to form for you the basic amino acid. which means that life, according to the scientists, started back then, billions of years ago maybe, based on the combination of these things. Okay? So yeah. Let's now talk about something called a hydrocarbon, which is basically made of carbon plus hydrogen. If you see a hydrocarbon that's made of only carbon and hydrogen, it's going to be basically nonpolar. It's going to be a nonpolar molecule. Okay? Some very common examples are going to be things like ethane, which is C2H6, propane, C3H8, butane, C4H10.
Are these the type of hydrogen carbonate?
These are the types of hydrocarbons because they only contain a hydrogen and a carbon. They tend to be nonpolar. That means they will not mix with polar. That means if you put those in the water, nothing will happen. It's like trying to wash the pan that you used to make eggs in the morning with water. It will stay oily because oil is made of these things, carbon and hydrogen. Okay, and those hydrophobic tails that we talked about earlier, they're made of hydrogen and carbon, and that's why they're going to be nonpolar. That's why. Okay.
They're
They're hydrophobic, exactly. They're going to be hydrophobic.
So every hydrocarbonate carbons, they don't mix with water?
Hydrocarbon don't mix with water. No, they don't. So here's an example of a fat droplet. So fat is made of fatty acids. And here we have 3 of them. Because there is a special fat called a triglyceride. And it's made of something called glycerol and also 3 fatty acids. And those fatty acids are going to be so nonpolar or so hydrophobic that again it's like getting an oil stain on your clothes. It's a big headache because with water they simply don't go away. Okay? So triglycerides have 3 fatty acids, but if you guys recall this phospholipid, it only has 2 fatty acids. So here we have the names of 2 hormones, both of which are going to be hydrophobic. How would you know? Because if you look at them, they're made of rings. And at the corner of each ring, there's going to be a carbon that's bonded to 1 hydrogen or more. So right now on this ring, you have so many carbons and hydrogens, it's going to be hydrophobic. So these things do not mix with the water. But because they're hydrophobic, they're going to have an easy time passing the what? The cell membrane. Because now they can interact easily with the hydrophobic interior of the cell membrane. And they can now pass easily.
But we can know they're hydrophobic because of the CH3 and the OH. It's all hydrogen bonds.
First of all, there's an OH, which means there's a hydrogen bond. But it's only 1 hydrogen bond compared to maybe 20 carbons. So sometimes there's a hydrogen bond, but it's going to be outnumbered by how many carbons you have. Does that make sense? Right now I have 2 OHs here. That means I have 2 hydrogen bonds, because OH means a hydrogen bond. But count how many carbons you have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Those 18 carbons will outnumber the hydrogen bonds, making the whole thing hydrophobic.
So we should just know in every corner there's a carbon.
You're right.
And a hormone?
in any ring. If you see any ring in biology, that means there's going to be a carbon and also hydrogens in every corner. In this case, we're going to have 1, 2, 3, 4, 5 carbons along with multiple hydrogens.
Okay.
Now let's talk about some functional groups. So there is something called a hydroxyl OH, which we just saw earlier. And whenever you see an OH, that means you expect there to be a hydrogen bond, essentially. For example, here we have ethanol, which is going to be an alcohol, which has an OH in the end. That means it's going to be having an easy time mixing with water. Now, of course, it does have carbons and hydrogens, but only 2 carbons. So ethanol is going to be polar. Does that make sense? It only has 2 carbons and 1 OH. So it's polar. But if you guys look here, earlier, we had 2 OHs and we also had like 20 carbons. So of course, in this case, the carbons will outnumber the OHs, making the whole thing hydrophobic. But if you just have 1 OH, for example, and only 2 carbons, that's polar. It can mix with the water. We also have a carbonyl group, which is made of a carbon with a double bond to an oxygen. Okay. A carboxyl group is going to be very important in biology. It's going to be a carbon double bonded to an oxygen and single bonded to an OH. That's found toward the end. Okay, toward the end. And so here, for example, toward the end of the molecule. Vinegar has this carboxyl group. And vinegar mixes with the water pretty well. So why so? Because here you have an OH, that's a hydrogen bond. Yes, you do have 2 carbons, but only 2, not so many. So
it's polar?
So it's polar, exactly. Amino group, NH2. It's found in amino acids. By the way, carboxyl group and amino group are both found in all the amino acids, and we have 20 of those. They must all have those 2 groups, basically. One at one end and one at the other end. For example, here we have the amino group at one end. We also have the carboxyl group at the other end.
So is this gonna be polar?
So the amino acid, polar, non-polar, it depends on something else, not on those 2 ends. It depends on what you see here sticking out. Okay, so right now here it's a hydrogen, but it can be other things as well. So to know,
it's hydrogen, it's gonna be
non-polar. Because all the amino acids have this, so cancel it out. And this, cancel it out. And also this part. What remains is the last part here. It can be polar, it can be non-polar. In this case, glycine is non-polar. We'll talk about that too. Here we have cysteine, which is also an amino acid. How do I know? Because I see a carboxyl at one end and I see amino group at the other end. But here the functional group is going to be sulfur, or as we called here sulfhydryl group. So if you see SH, that's called sulfhydryl group. Only 2 amino acids have that functional group. Only 2. One of them is going to be called cysteine. Okay. And this is called a methyl group, CH3, which is nonpolar.
CH3?
Nonpolar. Phosphate group, polar.
But isn't CH3 hydrogen bond?
No. You see, hydrogen bond only happens when there is hydrogen and only one of 3 elements. Oxygen, nitrogen, fluorine. Not carbon.
Okay.
Oh, by the way, you will see this phosphate group in multiple things, including phospholipids, DNA, RNA, and also ATP. Okay. So ATP is going to be very important. It's the source of energy for the body. If you go for a run, if you go for a walk, if you just climb the stairs, you have to use your ATP. ATP, of course, comes from food. So you have to feed yourself to get the ATP to make your body do the job. So why is it called ATP? Adenosine triphosphate. So it must have 3 phosphates. And all of them have a negative charge. And they're so close to each other, which is against chemistry. Chemistry doesn't like this many negatives close to each other. That's why ATP stores lots of charge. And once one of the phosphates leaves, you will get lots of energy released. Lots of energy. So right now, if you react ATP with water, a reaction that's called ATP hydrolysis, you will lose one of the phosphates. So you will only end up with 2 phosphates, which is now called ADP, with the phosphate that left with energy for you to do your work, like walking or running or something like that. Okay? So
what are the P?
the P is
The yellow
phosphate.
ones.
Yeah. So these individual ones are called P's. So each one here, if you look back here, that's a P. And if you have 3 of them, that's triphosphate. 2 of them, that's diphosphate. Make sense?
Yeah.
Okay. All right. Now, there's something called a polymer and something called a monomer. A monomer is going to be a single unit, while a polymer is going to be multiple units that have been put together, okay, in a covalent bond. So multiple units that are bonded by a covalent bond, okay. Those polymers will come in many forms. They can come in the form of carbohydrates, nucleic acids, proteins, okay. All of these forms are possible for the polymers. Carbohydrates are basically found in bread, in potato, in pasta, and also in rice. They're basically your energy resource. If you're hungry, you're gonna go and eat one of these things. The monomer of a carbohydrate is called a glucose. So the single unit of a carbohydrate is called a glucose. Now, if we're discussing nucleic acids, which are found in your nucleus, the DNA, or cytoplasm. So look, nucleic acid can be DNA, or it also can be RNA. If it's DNA,
These are types of polymers?
of nucleic acids.
No, the...
Oh yeah,
all of these
yes, carbohydrates, nucleic acids,
proteins.
and proteins. In the case of nucleic acids, we have 2 types, DNA and RNA. DNA never leaves the nucleus, but RNA is made in the nucleus, then it has to make its way to the cytoplasm. Okay. Proteins are going to be the 3rd type of polymers which have multiple functions, multiple. Okay. The monomer for them is called the amino acid. And by the way, the monomer for the nucleic acids is called a nucleotide. Okay. Now, lipids are also made of many things, but they're not called polymers. Okay. But we'll also talk about them. So one of the examples of lipids is going to be a phospholipid, which is made of a polar head with 2 fatty acid tails, which is going to be an integral part of the cell membrane to regulate what's entering and what's leaving the cell. Okay, so macro for big, right? It's the opposite of micro. They're built from monomers. All right, so a polymer is made of many monomers that have been put together by a covalent bond. Okay, now if you're trying to break a polymer down, you must use an enzyme. So an enzyme will make the reaction faster. How does it do so? You guys know? By lowering something called the activation energy. It's going to make the reaction faster. If you're trying to connect a monomer to another monomer, that will be called a condensation reaction. And the bond between them is called a covalent bond. And by doing so, you will lose a water molecule. That's why it's called a condensation reaction, because you will lose a water molecule. Okay? And so yeah, it's also called a dehydration reaction. So whenever you connect two small things to form a larger thing, you have to lose a water molecule. You just have to. So
This is not related to enzymes, right?
no, this is just part of the process. Right? So water molecule H2O is made by an OH from one of them and H from the other one. So one monomer gives up an OH, other one gives up an H, combining to form a water molecule. Now once you have formed your polymer, if you want to break it down, you now have to add water, called hydrolysis. So anytime you see hydrolysis, that basically means adding a water molecule. And that way, you can break the covalent bond and you can get your monomers back. Okay. So, adding water breaks, removing water is for making or forming. You see, this is going to be a hydrolysis reaction because you're adding water. And if you are making water, that means you're doing a dehydration reaction with one monomer contributing a hydrogen, other one contributing an OH.
So if you're adding H2O, it's hydrolysis, and if you're removing it, it's dehydration.
Yes. So what is the importance of carbohydrates? To give you fuel so you can do your work. So a sugar is a carbohydrate. Right now, if you go and buy, let's say, M&Ms, that's sugar, right? That's carbohydrate. If you eat bread, that's carbohydrate. Now, we have three different classifications for these, right? We have a monosaccharide, we have a disaccharide, and we have a polysaccharide. A monosaccharide can be, let's look here. Yep, a glucose is a monosaccharide. A galactose is a monosaccharide. A fructose is a monosaccharide. As you guys can see, all of them are made of the same chemical formula, C6H12O6. So they have the same formula, but they're built differently. This is called isomers. So same formula, but simply a different structure. You guys can tell, right? Like here, for example, in the case of glucose and galactose, there's a carbonyl, right, in the first carbon. But then look at the third carbon, one has OH on the right, one has OH on the left. This one difference changes everything. Okay. Look at fructose, the carbonyl has now been moved to the third carbon. Second carbon, not the first. This also changes everything. So different structure means different function. They're all called monosaccharides. Okay, they're used to build disaccharides. They're also used to build polysaccharides. Okay, but the main one is going to be glucose. That's your main focus. Glucose is going to be the center of life in biology. Whenever you eat something, it has to become glucose at some point if it's a carbohydrate to give you energy. So this is a glucose molecule right here. This is called the linear form. So C6H12O6. But it can also appear as a ring form, which can also be written like this. So if you see this ring form, don't freak out. It's just a glucose, a single glucose. Okay, so glucose is going to be so important. It's going to be the raw material for the process of cellular respiration, which is the process by which cells extract energy from the glucose molecule by breaking it down inside your cells. And the carbon skeleton of the glucose molecule will also be used to make other things. Okay, other things like fatty acids and also amino acids. That means you are able to get proteins and fats from eating carbohydrates. It's possible. A disaccharide means we have two monosaccharides that have been joined by a covalent bond. To join things, you have to have a dehydration reaction. So maltose here is going to be an example of a disaccharide. It's the result of joining of glucose with another glucose, with one glucose contributing an OH, other one losing an H to form a water molecule. And the bond between them now is a covalent bond called glycosidic linkage. You can also make sucrose, which is another type of a disaccharide. How? You can do so by joining a glucose with a fructose while also losing a water molecule.
But you can't do it if you want to gain a water molecule. Hydrolysis.
Hydrolysis,
Yeah.
you have to add a water molecule.
Yeah.
Right now we are gaining one. That's why it's called dehydration. So look, right now if someone is dehydrated, it's because they have lost their water. Right now we're losing the water. So to build a bond, you lose water. So essentially, to build a bond, you have to lose water. Okay, polysaccharides, from the name, they're made of many monomers. Many monomers combined. Okay, which means many covalent bonds, which means many water molecules that have been released. Okay. Now, those polysaccharides come in different forms and also in different functions. For example, if you're discussing a plant, it can be a starch or maybe a cellulose. But if you discuss an animal, it's going to be a glycogen. So humans and animals both have glycogen. Starch is used for storage, just food storage, while cellulose is used to build a cell wall, basically.
It's used or is is it used to make?
It's the component of the cell wall.
Oh. Okay.
Yeah. So we don't have cell walls as humans, but plants have. And their cell wall is made of cellulose.
Oh. Okay.
Yeah. You see, a potato that we eat has lots of starch, for example, right? But a cell wall has lots of cellulose. Okay. Animals and also humans we store our extra monosaccharides, our extra glucose, as a glycogen, basically. So a structural polysaccharide is not the same thing as a storage polysaccharide, because they're only used for holding the structure together, right? So we have a skeleton, we have bones, plants don't. And instead they have a cellulose to hold them together, to keep them tough.
Oh, but we don't have it.
We don't have cellulose, we don't have cell walls. We have skeletons instead. Now, glucose is used to make starch, and it's also used to make cellulose, but not the same type of glucose. Alpha glucose is used to make starch, beta glucose is used to make cellulose. Do you have to know that? Of course not. Just know that not the same glucose is used to make both. That's why they have different properties. Okay. So, for example, in the case of starch, as you can see, all the OHs are situated downward. But in the case of cellulose, you have down, then up, down, then up. So you essentially have different arrangement. You have different shapes, which results in different functions and also different structures. Yep. Okay, so only a few organisms possess the enzymes that are required to digest cellulose. So right now we cannot. So humans don't digest cellulose. But if you take a cow or a sheep or a goat, they could. Because they have microorganisms that live in their guts that do that job that we don't have. Okay? So some microorganisms that live in the guts of some organisms, can digest cellulose, breaking it down to glucose and giving them food, like a cow basically. Okay. So why would those microorganisms do this favor for a cow? What are they getting in return? Food. Some goes to them, and they also get to live safely inside the cow's gut, right? Yeah. For example, if you take a termite, which is like a small ant, it cannot digest the cellulose that it eats. It actually eats wood, but it cannot digest it. However, it has prokaryotes that live in it that can do the digestion for them, which makes wood for them a meal, essentially. Okay? Right now, if you just go and bite on wood, you won't get any benefit, but they will get lots of benefit thanks to their microorganisms. We also have another polysaccharide that's called chitin, which is found in the layer or the shell of an insect or a spider or a crab, right? To put their shells. That's why, for example, if you right now step on a cockroach, you're gonna have a hard time killing it, right? Because of the chitin.
Chitin is the top layer of them?
The top layer of the shell. Yeah, it's like it makes it hard. So it's hard for us to actually kill them.
Oh, that's why they do a little crunch when they die.
Yeah, lots of crunching. That's if they die. And also we have chitin found in fungi. Okay, it's their polysaccharide used to build their cell wall. So you know how plants use cellulose? Fungi use chitin instead. Okay, lipids. Lipids, for the most part, will be nonpolar. For the most part, will be hydrophobic. They barely mix with the water. Like I said, having an oil stain is not a good thing because you're gonna have a hard time taking this stain away from your clothes. You must use soap or something like that. If it's just water, it will remain on your clothes. Okay, so if we think of a fat molecule, what do we think of? We think of two things: glycerol and also the three fatty acids. Okay, and they must be joined by three bonds. That means you must make three water molecules. Those fatty acids tend to be very nonpolar. They're made of 16 or 18 carbons. That is so nonpolar, they will never mix with the water. Okay. They're called fatty acids for a reason, because they have a carboxyl group at one end. So they have this group at one end basically. And the rest is going to be a hydrocarbon chain made of lots of hydrogens and also carbons. If you want to join the fatty acid to the glycerol, you have to lose the water molecule by a dehydration reaction. Okay, you see, this is the glycerol. It's a 3-carbon molecule. And you must join it to 3 fatty acids. At each point of joining, you must lose one water molecule. You must join at 3 points. That means you have to lose 3 water molecules, basically. Okay, that means you must have 3 dehydration reactions upon making the fat. And the bond that will link the glycerol to the fatty acid is called an ester linkage. You must have 3 of those for every fat, for every triglyceride. So this thing here is called a triglyceride.
Are each those fatty acids?
Yes. The ones that are in yellow are called fatty acids. Now, fatty acids can come in 2 ways. They can be saturated, they can also be unsaturated. If they're saturated, they're made of single bonds, right? Like this one and this one. But if they have a double bond, they're called unsaturated, which will cause it to bend, as you guys can see. The molecule will bend. So this double bond causes bending. This is called an unsaturated fatty acid, which will make the fat liquid. Okay, for example, a vegetable oil has lots of these, which is why it's liquid. But right now, if you guys look at butter, it has lots of single bond, not double bond, because butter is solid. So right now, if you're looking at, some vegetable oil, like olive oil, it's going to be a liquid form because unsaturated. But if you're looking at a butter, that's all single bonds in the fatty acid, saturated, you're going to have it to be solid at room temperature.
So saturated fats are solids?
Yep.
And then unsaturated is liquid?
For the most part. I'm going to show you an exception very soon. But for the most part, they're going to be liquid, yeah. You see, they're going to be liquid if it's a cis double bond. If it's a trans double bond, solid. Get it? So a cis double bond.
What's that?
Like this.
Like that?
Yeah.
The circles?
I'll show you what that means. So look, right now, if you have this thing here and this thing here, and if you have here, let's say CH3 and here CH3, that's called cis. Why? Because both CH3s are to the top, or let's say both are on the bottom. But if one CH3 is on the top and one is in the bottom, that's called a trans now. and a trans double bond is going to be solid at room temperature.